All right, biology students, we are going to talk about protein synthesis and codon practice today. So we need to go over some basic things. So that means I'm going to need the highlighter. Aha. All right. Now, let's go over what protein synthesis actually is and how codons are related to it. So protein synthesis is the process where we sequence our DNA. We use it to build a protein for an individual amino acid. So remember that amino acids are your building blocks or your monomers for your proteins, and we're getting this from our DNA. Remember, the word synthesis means to build or create, so we're making proteins. The first step of this process is called transcription. So this is the important first step, and this is where we're going to go from our DNA and it gets converted into RNA. So not all of our DNA, just a portion of it. If I only need to create skin cells, I don't need my entire DNA. I just need my genetics that code for my skin cells. So during transcription, mRNA or messenger RNA is made from DNA sequence following the base pair rules. So remember in DNA, our base pair rules were A and T. So let's make a little chart, DNA. Our base rules were A always paired with T and C always paired with G, or vice versa. But with RNA, it's a little different. So with RNA, we have a new base pair, uracil, which is going to be U. So A and U are going to go together. And then C and G are still constant. All right. And that's what this is saying here. The DNA sequence followed the base pair rule, except RNA does not contain thymine, but instead uracil. So mRNA leaves the nucleus and then goes to the ribosome, which is in the cell's cytoplasm. The ribosome reads the message 3 bases at a time. So that's important, and that's what's called a codon. So we're going to read things in 3. And you might be able to see a little further down, we have our first base, our second base, and our third base. And we'll talk about how to use both of these charts. But we also need to remember that codon is going to come in 3s or triplets. Each codon will specifically single an amino acid. So we have different types of amino acids. And the amino acids are joined together and folded into proteins. And this is what's called translation, where we get our nice protein at the end. All right. This is the general basis, very basic version of protein synthesis. So here are our key points. DNA is used to make a copy of mRNA. That's where transcription comes in. mRNA, or messenger RNA, leaves the nucleus and goes to the ribosomes. Ribosomes are cell organelles that make proteins. Makes sense? 3 bases or 3 letters is going to give me a codon. 1 codon is going to give me an amino acid. A chain of amino acids, because remember, amino acids are the protein's building block. They're their monomers. It's going to give me a protein. And protein synthesis is also called translation. It's a lot of information to keep up. So let's get some practice in. So biologists use a codon chart or a codon wheel. Now, I'm going to teach you how to use both of these because you never know on a test, state test, ACT, test in normal class, which one you're going to use. So we'll go over both. Some people prefer the square one. Some people prefer the circle one. That's kind of a personal taste. But you never know which one you're going to see on a test, which is why it's important to be able to read both. Amino acids are usually abbreviated, and this is how we're going to keep it. We're going to use the abbreviations. And let's get to it. So down below, we're going to be using the codon chart. Pull it up a little bit more. There we go. So down below, we're going to use the codon chart to write amino acids that correspond with each codon found in my mRNA. So we're going to do CCC. We'll start off with this one, and then we'll do the same thing with this. So notice how it has 3 letters. Remember, the letters are your bases. So we have C, C, C. First base is a C, second base is a C, third base is a C. So if we're using the square codon chart, our first codon base is going to be on the left-hand side. And what that's going to be is it's going to be the letter C, or cytosine. So I'm just going to highlight that real quick. So that means that this entire row is going to be an option. Then we're going to go up top where our second base is. Our second base again is C. So I'm going to highlight that. And now we have everything from here. So this was our first one. This is our second one. And I want you to notice where they overlap, which is this square right here. And then we have our third base. So you see, you can have U, C, A, or G. Well, our third base is C, so I'm just going to take my highlighter and go right across there. So the one where my first base hits, my second base hits, and my third base hits is right here. And that is pro for our amino acid. All right. Can you simply just look at it and then figure it out? You can, but that can be time consuming, and it's always good to know how to read. But you can also look and see, it doesn't matter if it was CCU, CCA, or CCG, it would still give me the same amino acid. So let's try it on our wheel. So the way the wheel works is we're going to go from the inside and work our way out. So same thing, I'm going to find my C. All right, so here's the C. This is in this slice of pizza. Then I'm going to go to my next one, which is this C, because we're excuse me. There it is. We're working our way out, and then I'm going to see to the third C. And that gives me the same thing, which is proline. So that's how you read both of those charts. So let's try another one. We are going to look at CAG. Now take a second and pause this video and see if you can find one on your own. Use the chart that you prefer, and then we'll get rolling. I'll do both of them again. All right. Grab a different marker, or at least a different colored pen. So we'll just roll with this. So we have CAG. So same thing. Our first base is going to start here. Our second base is going to be A. So I'm going to go to this block over here. So now I'm looking at this square, because this is where my first and my second base meet. And then I'm left with a G. So I'm going to go to my third base over here. So it's G. So it's going to be CAG. There it is. It's going to be GLN. Same thing over here. We're starting off with C, which is our first base. We're going to go to our second base, which is A. So shift over here. And then we are going to go to G, which is right here. So CAG. And that's the same thing over here. Alrighty. I hope this was helpful. The key to these is just practice. And then, of course, if you prefer the circle one versus the square one, you use what you prefer. But please be aware that either one can be on a test, because they're going to give you the same answer if you know how to read them correctly. So don't favor one to the point where you can't read the other one. But basically, that's how you read a codon chart. It's really not terrible. It just looks crazier than what it is. I hope you have a good day, and I will see you in class.